Hi guys, Dr. Gillard here. It is Monday. It is winter 2021. It is week 5. So this will be our first kind of lecture that I did ahead. It's actually really Friday. So I'm doing this one ahead for some of you to see if you want to try to get ahead and listen to it, and then we'll check in on if you want. You don't have to, but I'll still be there Monday at 7:40. But if you have questions, show up and ask questions. If you don't, sleep in or go do something else. See how you like this. All right, here we go. So where have we been? So remember we got fertilization occurred here in the ampulla, and we created a zygote, and that immediately split into a two-celled creature called an embryo. The embryo split and split, and remember each one of these is called a blastocele, or I'm sorry, a blastomere, or daughter cell, but it's better to call them blastomere. And the zona pellucida stayed intact, unless you're going to have twins. Then they split into 4, 6. When it gets to 16, it's a morula. Between 16 and 32 is a morula. And then the morula drops into the martini glass we talked about last week, into the uterine cavity, takes on uterine fluid, and it carves out a cavity within this solid ball. You can see the cavity right here. That's called the blastocele. And it also gets a new name called the blastocyst. Okay, then we said the blastocyst hatches from the zona pellucida while it's floating around in the embryo. And we talked about the first gene differentiation. Genes turn on and cause the cells to morph into two cell types. A trophoblast layer, which is around the outside here in green, and then an inner cell mass was this in the middle. And then more cell differentiation occurred, and we got two very important kind of pre-super stem cell layers. They're kind of pre-germ cells. They're not the germ cells, but the precursors of the germ cells. So we have on top, remember this was the north pole, we called the embryonic pole. We called this the AB embryonic pole here. And then we differentiate into our little inner cell mass differentiates into two layers, the embryoblast and hypoblast in pink here. We got some AVE cells we talked about and the importance of them. This will be the cranial portion where they find. This would be the dorsal portion. And what else do we say? Let's see. I think that's about it, right? That should bring us up to date. Actually, this is cranial. This is caudal portion here, because the dorsal will be up here toward the embryonic pole, and then the ventral portion. So we know some planes, right? We learned some planes now. So this is kind of the creature we have. I guess we went a little further. So we talked about implantation and the receptors that need to be working there. And then our little creature, our little blastocyst, it binded and was sucked inside the uterus. This is mom right here. This is the endometrium. And this is about the size of a pin, by the way. It's so super small. But that's the endometrium right here. And these crazy cells, remember the syncytiotrophoblast cells were derived from trophoblast cells. And they're like a cancer. They invade and pull the little blastocyst inside mom's uterus, or uterine wall. We talked about the hormones. HCG was secreted, which keeps the corpus luteum alive. So estrogen and progesterone can keep being secreted, which are important for development of this endometrium. And yeah, so that brings us to date, and we go back to the Game of Thrones. Remember when they were invading the wall here? So the syncytiotrophoblast will keep on invading and deeper into the endometrial tissue, like the army of the dead, right? Trying to invade the land of the south, or south of the wall. And by day 10 and 12, the embryo is completely swallowed by the endometrium. So here's a picture of that. So this is day maybe 12. Day 12. And yeah, our blastocyst is completely submerged here. This is the Martini glass, the uterine cavity out here. There would normally be a little scar right here, so you'd have a little bit of a plug that scars up. But yeah, and look at how the syncytial trophoblast has gotten huge, right? We could also see it's been tapping into these little capillaries, which we're going to talk about. So it's trying to get a blood supply going as well, because right now we don't have any. We have no blood supply. So and this is called the stroma, endometrial stroma. It's completely surrounded by stroma by day between 10 and 12. Then we have something. So we have a problem. So look at this is mom's blood right here, and mom has a army that doesn't recognize this. And we said that the HCG helps make this kind of give this a pass, so mom's white blood cells don't attack it, but it's getting bigger, and that's not enough. So we need another mechanism to kind of call off mom's white blood cells from attacking this, because this is foreign. Mom doesn't know who this thing is. And so another problem is the cells don't have any blood, right? How are these? I mean, here, this is you. This is the little human right here. That's the bilaminar disc in a weird position. But nevertheless, there's no blood supply. I mean, even the syncytial trophoblast cells don't have any blood. They're starting to tap into mom's capillaries and get a little bit of blood. So we need to overcome those problems. So how do we do that? Well, welcome to the decidual reaction. The decidual reaction. So it's basically a process designed to feed the growing emblast without blood. So we got to get some food, some nutrients to those cells of the blastocyst. And we need to call off mom's white blood cells from attacking, because HCG was not enough. So the reaction starts when the stromal cells of the endometrium start accumulating glycogen and fat. So they get really, really big. And yep, that's what they do. And so once the stromal cells get so big, a name change occurs. They're called decidual cells. They're still stromal cells. They're just big. Stromal cells. So yeah, so at this point the endometrium is now also called, after the decidual cells, it's called the decidua. So you don't really talk about the endometrium anymore because it's morphed, it's changed, and got these gigantic decidual cells there. What do these decidual cells do? Does that say deciduous? That should say decidual cells. I just noticed that. Decidual cells, what do? Well, they start to release their fat and glycogen. They dump it into the stroma, and so it diffuses around the side of the blastocyst, and it soaks into the blastocyst, mostly into the syncytial trophoblast. But those are the cells that are active in dividing and growing. So they need it the most. And that will keep the cells alive and keep them growing until we can develop a circulatory system. Well, what about the mom's immune system? How do we take care of that? The decidual cells also release special types of cytokines which attract a special type of white blood cell. And this special, we're not going to go any deeper than that, a special leukocyte, which is a white blood cell, it secretes something called interleukin-2. And we'll just leave it at that. Interleukin-2 is able to grant immunoprivilege to our little growing blastocyst. So mom's immune system leaves it alone because of interleukin-2 that is floating around the area. All right, so now we're growing, we need to make some more structural changes. So the first thing we need to do is talk about Heuser's membrane. All right, and there's two waves that are about waves of cells that are about to occur kind of in an inferior direction. And if you're a surfer, this is, especially if you're a goofy footer, oh my gosh, this is just, I would be so excited if I saw a break like that, and there's really, well, there's quite a few people here already. Finding a great break uncrowded, even back when I surfed back in the day, back in the 90s, 80s and 90s, when I thought it was crowded then. I went out there one time and just to look, and I was like, oh my God, there's so many people. Anyway, this is just a very, if you're a surfer, would give you goosebumples. But anyway, so around day 9, the hypoblast cells at the cranial and caudal end of the disc, they start undergoing mitosis. And when you go under mitosis, one cell stays the same, and one cell, you got a new cell. And it creates a bunch of new cells, and they don't have anywhere to go, so it pushes them down around the blastocele cavity. Picture is best here. So here is our hypoblast cells in yellow, and these cells at the corner ends, they start undergoing mitosis and making new cells. And these new cells get ratcheted downward, downward, downward. And pretty soon they go all the way around and line our blastocyst cavity here. And these are called Heusner's cells, and they form Heusner's membrane. It's like Heuser's cells. Heuser cells. There's some AKAs, of course. Exothalamic membrane is another one. Primary yolk sac lining. Parietal endoderm. I mean, that's Carlson. It's my favorite book, but then we're getting confused. The reason they say it's parietal endoderm because this is going to develop. This hypoblast is going to develop into the endoderm, so I see where he's coming from. But I like Heuser's membrane. And then we get a name change too. So once Heuser's membrane, these yellow cells form completely around here and line this new blastocele cavity, we don't call it the blastocele or blastocyst cavity or blastocele anymore. So we are now going to call it a primary yolk sac. Primary yolk sac. Now chiropractors got to watch out because Moore calls it something weird, like no one else does. Moore calls it the umbilical vesicle, and he argues it's not. We shouldn't use the word yolk sac because humans don't have yolk like other creatures do. And I see that, but I wish he would quit like inventing these words and just stay with the pack and stay with the group, because it makes it teaching this and it makes it learning and testing out very difficult when there's so many AKAs. I'm calling it yolk sac more so, but watch out for umbilical vesicle. All right, so here's just a blow up. We have Heuser's membrane here in yellow. I should have made that a different color, could have made that orange. And then this is now the primary yolk sac, right? And then remember, we already had our trophoblast layer. It changed names to the cytotrophoblast layer in dark green, and there's syncytiotrophoblast. And yeah, we got some cavities starting to develop in here, so we'll see what that's all about in a little bit. So about day 12, another layer begins to appear. So this is an important one, and this is called the extraembryonic mesoderm. Extraembryonic mesoderm. This is very, very important, but be careful because this is not one of the germ layers. There's an intraembryonic mesoderm. Or if you just hear the word mesoderm, you assume they're talking about intraembryonic mesoderm, and we'll come to that. So this is extraembryonic mesoderm. Where does it form? It forms between the cytotrophoblast layer, Heuser's membrane, and the amnion. Remember we formed an amnion? Let's look at a picture. Picture's best. So here's a little view. Here's our epiblast layer. It's kind of a coronal view we're looking at here, or an axial view you could say. Hypoblast. Here's the new cells that came down and lined our old blastocele. Now we're going to call it a primary yolk sac. These are Heusner's membrane, or Heusner cells. And this was our cytotrophoblast we just looked at in gray. And now look, we got a nice thick layer that just popped up here. And it's going to keep on pushing and pushing. It's going to form all the way, whoops, not right here. It's going to leave a space right there. It's not going to form there, but everywhere else it's going to invade and push this apart right through here. except this region right here. We'll look at that in a sec. But we got new tissue, so this is called the extraembryonic mesoderm. And we're going to split this in half here in a second. So here's a rant of mine. Oh my God, I just remember building this course and going through this. I'm like, these authors, what in the hell? You know, why can't they get together? And just an example of the craziness. So Langman, and the question is, this important extraembryonic mesoderm, where the heck does it come from? Honestly, they don't know where it comes from. Langman, Chiropractic Board Book, says it comes from yolk sac cells. So we don't even have yolk sac cells. He's probably talking about Heuser's membrane. Now that's the first time they used yolk sac cells out of the blue. And it also comes from cells moving through the caudal portion of the primitive streak. Well, that's ridiculous because we're still in week 2 and the primitive streak doesn't even form to week 3. So Langman, come on. Moore says it's derived from, quote, the vesicle endoderm, end quote. There is no word in Google that's the vesicle endoderm. So there's the other Chiropractic Board Book. They probably, I assume sounds like he's talking also about Heuser's membrane, the cells of Heuser's membrane. Carlson says it's the parietal endoderm, so that's Heuser's membrane, that's his AK for Heuser's membrane, and the primitive streak. That would mean, again, that doesn't occur to the 3rd week. So I don't think that can be, unless it forms more slowly. And maybe it forms in week 2 and week 3, but they say it's complete by week 2, so I don't think that's true either. Singh is just way off base. He says it comes from trophoblast cells. So, and he shouldn't even call them trophoblast cells. They should be called cytotrophoblast cells. And then Larson says it's hypoblast cells give rise to it. And, I mean, hypoblast cells gave rise to Heuser's membrane, so I think that's what he meant. And primary yolk sac. So they're just all over the place on this. So, you know, I just hope that the people writing test questions, I haven't come across one, because I do write test questions for the boards, you know. But I haven't, it was canceled. I'm going to be on this committee, this test writing committee. So, boy, I hope a question comes up like this, and boy, am I going to let out a piece of my mind if I get a chance to. But this is a mess, so I'm hoping the boards stay away from it. So what are we going to go with? We're going to say Heuser's membrane and the primitive streak, even though, because Carlson said it and Langman both said it, we're going to go with that. But I'm not even going to ask you that. All right, that's enough ranting. No more ranting today. All right, so now this picture's very cool. So we can see Heuser's membrane here in yellow. And we can see the trophoblast in green. And now we can see our new extraembryonic mesoderm has actually pushed its way all the way around. Let's see, is that correct? Yes, it is correct. So I was wrong. It doesn't split in this region right here. So it does completely encircle the little human right here. So this extraembryonic mesoderm completely encircles the bilaminar disc and the primary yolk sac. Right? But splitting, it's going to leave this area right here, which is going to ultimately be the umbilical cord. There's a real human picture of it. Bilaminar disc, epiblast, hypoblast, and you can see this is Heuser's membrane right here. Guess I can color it in, why not? Heuser's membrane. Right, there's the cytotrophoblast layer would be right here. You can see this one just got barely swallowed. Great, and then all this stuff in the middle, that's the extraembryonic mesoderm. Right, next thing that happens, kind of the same story when we were talking about oogenesis, we get some cavities right in the middle of that extraembryonic mesoderm. And pretty soon the cavities coalesce together. So we're basically splitting the extraembryonic mesoderm into 2 pieces. So an inside piece, which the word splanchnic comes to mind, and an outside piece, and the word somatic comes to mind. specifically somatic mesoderm, splanchnic mesoderm, more specifically extraembryonic somatic mesoderm, extraembryonic splanchnic mesoderm is right there. But notice now the splitting didn't occur, and this author's off a little. It should be more right about here where the splitting occurs, but it doesn't completely split all the way around is the key there. So the cavities coalesce. Those are called the extraembryonic coelom. Those little mini cavities, I don't think that's super important. But there's a very important AKA. Well, I mean, it is important actually. The coalesced cavities form something called the chorionic cavity, and that is the AKA, extraembryonic coelom. I was thinking about these in between. Oh, here they are. The extraembryonic cavities. These are little cavities. I don't think that's important, but I do think extraembryonic coelom and chorionic cavity is important, because pretty soon, see how it's going to completely split, and now it's formed a cavity. There's no more little subcategory or subcategories here, and it's going to keep ripping and ripping and ripping and ripping. All this pink is extraembryonic mesoderm, so it rips and rips and rips, and it stops right here, and that is going to be basically your umbilical cord. Right, so that's the story with that. This cavity, this is called the chorionic cavity, or the extraembryonic coelom, or the chorionic sac. So make sure you know what that is, because that word is going to be familiar. And you can see how in this picture it's advancing. It's going to stop right there, but this side is going to keep ripping and ripping. And the pink again, that was all extraembryonic mesoderm. But we see something else happening, don't we? Look at that. What happened to our giant secondary yolk sac? It's getting pinched, isn't it? So we don't understand how this happens, but this chorionic cavity somehow is pushing in here really strongly. And in fact, it's going to pinch this thing off right here. And all of this is going to degenerate away into nothing. And we're going to be left with what we're going to be called the secondary yolk sac, because the primary yolk sac is gone. So I just jumped ahead there, but yeah, we just created some new tissue layers. As I said, we have a splanchnic extraembryonic mesoderm and a somatic extraembryonic mesoderm. The AKAs are going to upset you, and I apologize, but there's nothing I can do about it. Some people call it, and I like this idea, because you're going to learn about, there's 2 layers around the heart, 2 layers around the lungs, 2 layers around the abdominal cavity. The 3 P's, pleural cavity, pericardial cavity, peritoneal cavity. And they all have 2 layers, and this is where these 2 layers come from. And there's those 2 layers. There's a parietal layer and a visceral layer. And some authors like to say visceral mesoderm, or extraembryonic visceral mesoderm. And I like that idea, but the board books don't tend to say that. They tend to call it splanchnic extraembryonic mesoderm and somatic extraembryonic mesoderm. And there's a lot of AKs, so there's all the different AKs. So board book, actually one of the chiropractic board books, I've seen them all of a sudden out of the blue just say visceral mesoderm. So if you ever see visceral mesoderm, that I like it, but they're not consistent with that term. But visceral mesoderm, and then parietal mesoderm, that's great. Visceral is always on the inside toward the viscera of the body, and parietal is always more toward the outside. So that is the story. They're found internal. So the splanchnic extraembryonic mesoderm is found internally to the chorionic cavity. What do you mean by that? Well, there's the chorionic cavity. So this is all splanchnic extraembryonic mesoderm, or you could call it visceral mesoderm, or visceral extraembryonic mesoderm. Once you learn your AKs, I know I'm probably sound like I'm talking a foreign language right now, and I think I am until you start to learn these words. But this one is the outside layer. So there's the parietal mesoderm, unfortunately also known as extraembryonic somatic mesoderm. The AKs will drive you crazy. As if this class isn't hard enough without all these damn AKAs. Okay. All right. And I said it already that this area is not going to split. So that's going to become the umbilical cord. Before the umbilical cord, it's called a connecting stalk. Connecting stalk. Okay, somatic, same thing. Visceral mesoderm, I just talked about this whole thing, so I won't go over that again. It's outside of the chorionic cavity, or roofs the chorionic cavity, if you will, and the amniotic cavity up top there. Okay, and let's see. So not all extraembryonic mesoderm splits. Yep, so this is that stalk right here, and you'll see that will become the umbilical cord eventually. The connecting stalk becomes the umbilical cord. And here's the chorion. We need to talk about that. The amniotic cavity, look how big that amniotic cavity, we haven't talked about that in a while, but that thing completely encircles the little human here eventually. All right, what is the chorion anyway? So that's another concept, that high yield board question you should know. It's basically, chorion is born after you create the extraembryonic somatic mesoderm, or that outer layer, or that, that AK would be the parent, the parietal mesoderm. So the combination of the extraembryonic somatic mesoderm plus the cytotrophoblast, that gives that double layered structure is called the chorion. And this releases some really important some gene factors, or some gene products that will help form the circulatory system here in a minute. So key fact right here, chorion is somatic mesoderm plus cytotrophoblast, that outer layer there. And it will someday encircle the entire secondary yolk sac, except for that connecting stalk again. Right? So this is the chorion. And the chorionic cavity is forming. It's still in that kind of infant stage. Pretty soon all these partitions will be gone. But here's the chorion. So it's this outer layer, so that's splanchnic mesoderm, or that, somatic mesoderm, aka parietal mesoderm. And then the light green right there, that's the cytotrophoblast. So those two layers. It's not the dark green, that's syncytiotrophoblast. I know, I feel your pain. What can I tell you? Dermatology is not easy. Or not dermatology, embryology. I always get those two mixed up. I just did a dermatology lecture right before this one. Okay. All right, secondary yolk sac. I kind of spilled the beans on this one. But that chorionic cavity is going to pinch that primary yolk sac in half and create a new cavity. So this is wave number 2 before this happens. At the same time, we get another wave of cells coming out of the hypoblast. This one really doesn't have a name. This is the one you could probably call yolk sac cells, I think. But it's the same kind of deal like Hauser's membrane was formed. And so let's just go to this picture. So again, cells right here in the hypoblast start waking up via mitosis, and they start making these pink cells. And they don't have anywhere to go, so it keeps splitting and splitting, and it pushes them down and down and down. But perfect timing, this chorionic cavity grows and it pinches off. It pinches the primary yolk sac in half. And as it does that, instead of these yolk sac cells going all the way down here, it pinches it off so it just surrounds this new layer. So these yolk sac cells, they are on top of this is Hauser's membrane in yellow here. They're on top of that, but they don't go down to this kind of port. Their south pole is here, the abembryonic pole. They don't worry about this. This is all going to degenerate away here. Right, so that's kind of the story there. What makes the chorionic cavity pinch this off? We have no idea. We don't have any genes. We just know what happens, and we just don't know what it is yet. All right, so yeah, so that new thing is the secondary yolk sac. So, and I said that's going to degenerate. There's the kind of the south pole part of that primary yolk sac. It's pretty much got much smaller. You see, this will be completely degenerated away here. And yeah, so we actually lost quite a bit, because remember what this is? That's that mesoderm right? That split. So that's the inner layer of mesoderm, which has all those AKAs. We can call it splanchnic extraembryonic mesoderm. But we lost a lot of that. This is all going to disintegrate, and that's all splanchnic extraembryonic mesoderm gone. All right, so we get a name change now. We can't call that primary yolk sac anymore. So they name that secondary yolk sac, and that's the degenerating primary yolk sac. All right, everything I say. Watch out again, Langman. Come on, Langman. Calls it something else. He calls it the definitive yolk sac. Definitive yolk sac. So there's another AKA for you. All right. Let's see. Extraembryonic splanchnic mesoderm disintegrates. Yep, we said that already. Everything I just said. And yeah, here's what we have now. It's completely degenerated away. Chorionic cavity is taken over. And that splanchnic mesoderm is, most of it is gone, but there's still some surrounding the Heusner's membrane here and the yolk sac cells, we'll call them in pink. And yeah, that's the story. Secondary yolk sac, definitive yolk sac will remain a major structure. And if you were a bird or an animal, this would be filled with yolk, and that's how you'd be getting your nutrients, because animals don't develop a blood supply so quickly like we do. But we don't have one. That was one of the board book's authors' arguments, that why call it a yolk sac when we don't have yolk? And I get it, but this is already a hard enough class. So I would just stay like with the other authors if I was writing a book like this. Anyway, yeah, so what does this yolk sac do in humans? It is a place that helps form blood vessels. Hematopoiesis occurs here in blood cells. It helps form blood cells, which doesn't form blood vessels, it forms blood cells. The secondary yolk sac lined with endoderm, Langman, okay, because the first and secondary primary yolk sac layers came from hypoblast, and hypoblast is going to morph into endoderm. Some people, like Langman, tend to call this lining an endoderm lining, which no one else does again. So I don't particularly agree with that. But let's go back to here so we can see what the heck he's talking about. Where were these? Where was that? Sorry, I'm going like way back. Oh, here we go. So here is the hypoblast layer here in yellow. This is going to morph into one of the major stem cells called germ cell. This is going to be a germ cell layer. And Heusner's membrane comes from this, so he's saying that these can be considered endoderm cells. And the next layer that comes down, remember there's two waves, I'm calling them yolk sac cells. He's saying that can be called endoderm as well because it was derived from the endoderm. I think that's what they're trying to say there. I could probably throw that slide out because it's confusing. Oh man, we went way back, didn't we? Yep, okay. A formation of the connecting stalk. Atlantois, I took this out because that's a week 3. We'll look at that later. But I will talk about the connecting stalk again. Remember the amniotic cavity stopped right in this region here. About day 13, chorionic cavity has split everything except that region. That will become the connecting stalk, and it will become the umbilical cord later. This is nice though, a good anatomy question, or embryology anatomy type question. What is suspended from this connecting stalk? Because it's almost like an apple, right? The little blastocyst is an apple. What is suspended by the connecting stalk? Well, the bilaminar disc is. So hypoblast and epiblast are. The secondary yolk sac is. I mean, what's the parts of the apple? The amnion and the amniotic cavity. So those are all the stuff that is suspended. And we could add amnion in here too. And the amniotic cavity. Bilaminar disc, secondary yolk sac, everything I said. All right, so let's talk about hooking up the blood supply, because I mean, we don't have a blood supply and we have tissue that's growing like crazy. So back note, so even before full implantation occurred, this cytotrophoblast was developing these cavities. They're called trophoblastic lacunae. Lacunae is like a lagoon, I think. Or you could look it up, but it's like a little place, little cove in the lake. But that's what these are. They're little coves, and they don't have anything in them yet. But the cytotrophoblast is going to tap right into mom's blood supply here, and these guys are going to fill up with blood. And great, so all the blood can diffuse, and there's probably you can just simple diffusion can feed all the cytotrophoblast cells. But we still got a problem, because I mean, there's the human right here. The bilaminar disc needs to be fed, and so we're going to tap into those lacunae and grow blood vessels and blood cells to feed everything. And that's where we're going to go with that, or this story. But first, we have these trophoblastic lacunae. So once those lacunae form, the syncytotrophoblast cells somehow know where mom's blood vessels are, and they drive right into them, and they fill those lacunae up with blood. And so cytotrophoblast is very happy because it has blood in it now, but the bilaminar disc has nothing, so it's not happy. So here's a cartoon of the cytotrophoblast in purple has tapped into mom's capillaries, and all those lacunae are now filled up with blood. And I mean, this would be into the plane of the page. The capillary would be in the back, because remember, this is three-dimensional. So this would be going on everywhere, but all these blood-filled lacunae pop up now. And yeah, so that is pretty cool. The syncytotrophoblast is fed, at least. Bilaminar disc is not, and it's getting bigger, and we need to feed it. So now we need to develop a blood supply to the bilaminar disc. Remember the chorion, coming back to the chorion. So that was cytotrophoblast, the light green layer, plus the outer layer of that split mesoderm, which you could call the somatic mesoderm, the extraembryonic somatic mesoderm, the somatic extraembryonic mesoderm. What else can you call it? The parietal mesoderm, all those damn AKAs. But this guy right here, together these two were called the chorion. Well, so what? So what is this chorion is going to start forming a point, and it's going to stab into this lacunae and try to tap into that blood supply. And that is exactly where we're going. And that first little spike is called a primary chorionic villus. Villi is plural, villus is singular. And that's exactly what it does. Its job is to find the trophoblastic lacunae and connect with them. Right? And so that's exactly what we have here. We have a little point that's happened, and it's pushed into the lacunae. Well, great. So what happens next? So about the third week now, we're into the third week, the center of the primary chorionic villus fills with a new tissue, and it's a mesenchymal tissue. And we won't talk about the genes turned on, or because they don't even know the full story. But a magical mesenchymal tissue appears in the center of that little projection, that little point. And this is mesenchymal tissue is very close to stem cells. It knows what's going to make, though. You can't use it to make a liver or eye or anything else. But nevertheless, it's mesenchymal tissue. So once that mesenchymal tissue shows up, we get a name change, and the primary chorionic villus, or villi plural, becomes the secondary chorionic villus, or villi. Right? So here's a cartoon of, so we got this new green stuff growing in the chorion, and that is the mesenchyme that popped up. And you can see it everywhere. And so that's magic tissue, and it's starting to poke into these lacunae to get blood going there. But it doesn't do anything yet, because we need to make blood vessels, but that's exactly what it's going to do. So next, the mesenchyme in these newly formed secondary chorionic villi, the mesenchyme will morph. It'll differentiate into two things: a microcirculation, tiny blood vessels and blood cells. So this is a process called vasculogenesis. So this is the very first blood supply that is going to feed the bilaminar disc, which is the little human. Let's see how that happens. So more specifically, mesenchymal tissue morphs into these blood cell precursors and blood vessel precursors called hemangioblasts. Hemangioblasts. We'll do a little bit of molecular biology. We know that something called FGF2, fibroblast growth factor 2, which is expressed from cells of the chorion itself, it soaks into those mesenchymal cells and tells it to morph into this hemangioblast, which is kind of like a mesenchymal cell, even a little more limited though, because these hemangioblasts can only make two things. They can make red blood cells, white blood cells, lymphocytes, things like that, and they can actually make endothelial cells which hook together like crazy to form a blood vessel. So that's what hemangioblasts have the option to become. Where did hemangioblasts come from? Came from the mesenchymal tissue. This green stuff right here, that's what morphs into hemangioblasts. Why does it morph into hemangioblasts? Because it's exposed to FGF2. Got it? So now another one, so be careful, don't get these confused. Now we have VEGF, vascular endothelial growth factor. So the cells of the chorion, we won't get any more deep in that. Cells of the chorion also release VEGF2, and VEGF2 soaks into the newly created hemangioblasts and causes some of them to morph into endothelial cells, which form a tube, right? We'll talk a lot about endothelial cells in fifth quarter. They're amazing cells that form your blood vessels. They secrete so many things like nitric oxide, endothelin. They express LDL receptors. They're von Willebrand factor. There's so many things that they do that we'll talk about in fifth quarter. But yeah, and then some of the hemangioblasts get different genes on. So we got tubes made, but not all the hemangioblasts turn into tubes. Some of them have different genes that get turned on. We won't worry about it. But they turn into your first blood cells, like your red blood cells and your platelets and things like that, to get you started. And yeah, there's some gene that we don't know much more the story than that, but we know that the VEGF is the key for making these first red blood cells. All right, so here's kind of an overview. So there's the chorion, and of course the chorion would have all these cells here. And the chorion first secretes VEGF2, and that causes, there's VEGF2, we won't worry about the receptor, but it causes those mesenchymal cells to morph into hemangioblasts. And then the hemangioblasts, these are the hemangioblasts here, via VEGF and different receptors. I don't want to get that deep in it, but there's two different receptors that these can bind. But they can morph into endothelial cells to form tubes, or they can morph to create these blood cells. Right? So VEGF causes some hemangioblasts to morph into blood vessels and others to morph into blood cells. We'll just leave it at that. Once you have real blood vessels and real cells in those little finger-like projections, your secondary chorionic villi, they get a name change, and now they're called tertiary chronic villi. And it's the tertiary chronic villi that will tap into the syncytial trophoblastic lacunae, which have already tapped into mom's blood supply. Bottom line is we have blood now flowing through the syncytial trophoblast into the kind of the deeper meat of the blastocyst. So, and vasculogenesis will keep on going until pretty soon this is what we have. So there's these tertiary villi that vasculogenesis just goes and goes and goes, and pretty soon it goes all the way down the connecting stalk, and we have a blood supply around our little trilaminar disc here at this point. Right now we still have a bile. We haven't talked about forming the trilaminar, but we will. So yeah, that's the story. The uroplacental circulation, so once the blood is flowing from mom into the little blastocyst, we have formed what's called the primordial, the very first circulatory system. There's no heart yet, but we're using the mom's heart really to power blood through this little blastocyst. All right, so everybody's happy. So this is an important slide to study. We've just finished the week of twos. I mean, we've snuck ahead a tiny bit into week 3, but here's the key facts about the week of twos. Second week is the week of twos. So we took an inner cell mass last lecture, turned on some genes, we explained that, and we created an epiblast and hypoblast. We took the trophoblast and differentiated that into a cytotrophoblast and syncytiotrophoblast. Huge testing. Look at all the stars here. This is one of those things. You want to know what to study? Study this slide. We took the extraembryonic mesoderm and split it by making a chorionic cavity, which created 2 new layers. A extraembryonic somatic layer and an extraembryonic splanchnic layer. It should be mesoderm though, right? And that's got all those AKAs. We also formed 2 new cavities on our little journey in week 2. An amniotic cavity, primary yolk sac, really 3, and then a secondary yolk sac. So that one kind of breaks the rule because we really went through 3 cavities. And then we talked about 2 waves, mitosis turning on in the hypoblast cells, that created 2 waves and 2 new tissues called Heuser's membrane. And some unknown cells created a secondary yolk sac. We can call them yolk sac cells if you want. Some authors do call those yolk sac cells that second wave that came down and covered Heuser's membrane. All right, so there was a few more slides on here. I took them out because this is a juicy lecture and your heads are probably spinning around right now. But welcome to embryology. You got to know this stuff. So, all right, email me those questions and see you guys later.